Okay, good afternoon, everybody, and welcome to our first webinar this year. Today, we're going to be talking about variant classification and interpretation and the guidelines and how to use use of the various databases associated with it. Just got a couple of slides, the usual formalities, I'm afraid, but important that we go through them. So next slide, please. So a few housekeeping things. We have automatically muted all your microphones to avoid any background noise. We are recording the webinar, and as normal, we will share this with you via the GenQA and EMQN YouTube channels, usually 2 to 3 weeks after the recording's taken place. But most importantly, this is an interactive session, so your participation is also important to us. We have a Q&A session at the very end. And our experts are on hand, so please do use the Q&A session to log your questions, and myself and my co-panelist Sandi Deans will be putting those questions to the experts at the end of it. Next slide, please. And lastly, feedback is also important to us because we do utilize your feedback to improve our webinars. Over the years, we've introduced a lot of different things as a consequence of your feedback. So please do contribute to the feedback. You'll get a link through it via email shortly after the webinar has taken place to complete a short feedback survey. It usually takes no more than 2 minutes of your time. Well worth the effort. Please do contribute to that as well. Okay, that's enough of the formalities. I'm now going to hand over to my co-chair, Professor Sandi Deans.
Next slide, please. Thank you, Simon. And I just join welcoming you to the first webinar of quite a few for 2025. So it's with great pleasure that I introduce our two experts who are joining us today to share their words of wisdom and give us some guidance on how to classify our variants. We are joined by George Burghel from Manchester in the UK and Andrew Muranyi, who's actually from Edmonton in Alberta in Canada. So next slide, please. So this is just the disclaimers, and there's no non-promotional webinar organised and funded by AstraZeneca and MSD. Next slide, please. So I'm now going to hand over to George to kick us off. So today we're going to be covering variant classification and interpretation, specifically around guidelines, applying the guidelines and use of the data sets that are available, CanVig UK, CanVar UK. And then Andrew is going to take us on a review of NOMAD, ClinVar, ClinGen databases for use in variant interpretation. So George, it's a pleasure to hand over to you now to take us through your presentation.
Thank you very much, Sandi. Can you hear me okay?
Loud and clear, thank you.
Perfect, thank you. So I will be covering some of the resources. Our focus today between me and Andrew are covering some of the resources that are available. So I'm not going to go through any of the guidance. I will direct you to where you can find some of these CanVig UK guidance and also other resources that are available and how to navigate around the CanVar UK database. So this is the link for the CanVar UK databases available at the bottom of this slide. Many of you, especially those based in the UK, will be very familiar with this resource, but many outside the UK are less familiar. And we see that when we get the results from the schemes and running these classifications. So once you get to the landing page, there are plenty of resources that are available, and this page can navigate you around. So we will briefly look at the guidelines that are available under the consensus and under the gene-specific recommendations. We will also look at the CanVar UK variant database, some of the CanVig UK resources and the functional studies scoring. So all of the different sections that I will be presenting in this webinar, you can get to through this main landing page. So under the CANVIG UK consensus specification tab, you will find our generic guidelines. So that's the consensus guidelines for all cancer genes that do not have a cancer-specific guidelines. So for genes that are covered usually by this webinar, like BRCA1, BRCA2, and ATM, we do have gene-specific guidelines. But for other genes, you can find the consensus specification on this page. Also importantly, to familiarize yourself with this, if you are using the CANVIG UK guidelines, even if you're using the gene-specific guidelines, because the gene-specific guidelines will refer to the consensus guidance in many instances. There are also important specifications or guidance. Recently published the reduced penetrance specification. It's worth familiarizing yourself with that. It does include some relevant information, especially for example, BRCA1 and 2. And there is also a framework of what we can do, recommendations of the labs on what to do when there is a reclassification. So if a variant has been reclassified, especially when it crosses a clinical actionability threshold, what would we recommend from the CANVIG UK on what are the best steps to handle the reclassification? If you navigate back to that main page and then go to the gene-specific recommendations, you'll be able to access the guidance that handle gene-specific elements. And there are multiple of them available. I highlighted the relevant ones here, ATM, BRCA1, and BRCA2. And other than accessing the CANVIG guidance here, you can also link directly to the ClinGen VSEP guidance. And if you open any of these guidance, you will notice that in many instances, we try and align as much as we can with the ClinGen VSEP. And we make that clear. If we are aligning with a published ClinGen VSEP for that gene, we will make it, we will state that in the guidance section that this is, for example, in alignment with the Enigma BRCA1, BRCA2 in this case. But there are some instances where the CANVIG UK guidelines, the gene-specific CANVIG UK guidelines, can differ from the ClinGen VSEP recommendations. And there are important reasons for some of these differences. And one of the examples is listed here. So for example, for PS4 case control analysis, because in the UK we have the NHS setup, the National Health Service, we are able to collect results and variant information from affected patients that have been tested for multiple genes, including BRCA1 and BRCA2. And that data is collected nationally and then displayed on Canva UK. So accessing that data will give you a number of affected individuals with the variant, if there are cases, and with a denominator of the total number of affected people that have been tested. So does this actually provide us with a case set that we can use against a relevant control set, for example, a subpopulation of the UK Biobank and so on. So it is worth looking at some of these differences because there are reasons why the CANVIG guidance can differ from the ClinGen VSEP. So once you go through the guidelines, you will see the elements that are in common, but also you will see the elements that are different and the reasons usually why they are different. Other than the CANVIG guidance that we looked at, there are some also resources that help and support the process of the variant interpretation and reporting. So you will see that we have report templates, and they affect or they cover multiple genes. So it's the way that in the UK we recommend, for example, reporting variants of that are pathogenic or likely pathogenic and variants of uncertain significance from the different genes. But there are also specific instances that align with certain UK-based guidelines. So if you are based in the UK, I highly recommend looking at some of these. So for example, ATM, we have restrictions on what we report in the UK, and so we make specific recommendations for some of the variants. So it's worth familiarizing yourself. If you're in the UK, we recommend following these. If you're outside the UK, we're hopefully giving you some useful templates that you can use to impact your local practice. There are also other resources. Again, some of these are very UK specific. So for example, because of the restrictions that we have on the reporting of ATM and CHEK2, for example, limited mainly to truncating variants, there are exceptions to these rules, and we put a framework of what people can follow if they find a variant that doesn't fit within the current guidance and whether they should report it or not. Again, it is useful for colleagues from outside the UK to explore. But there are also training resources available. So every month we have a CanVig meeting, and during the CanVig meeting, we usually often provide some training and education. And all of these training resources, the slides, the presentations, are available to explore and download from the CanVig website. Now, if you go back to the main page and click on the CanVar UK database, you'll be taken to this gene and variant database, which I think is really important. And we, again, heavily use it in the UK. It is available for international colleagues, and many people don't know that. And other than the open access that you have, we recommend that you register to the CanVar database. So if you go to the top right corner, and you can register by emailing the CanVig ICR team. And that's really helpful because, as we will see in the next few slides, that you can access a little bit more resources that are not available if you are not registered. So currently there are 1,292 members. This is data provided from the CanVar team. And although mainly, as you can see in the pie chart, UK based, we do have quite an international variety of members. And I think it's really important, as we will see in the next few slides, for international colleagues to interact with these resources. And there are ways that you can communicate with each other using the CanVar database. So once you're in the database, you can explore the genes that are available. So you can choose one of the currently 116 genes available, and then you can add your variant, and then you can explore various things. Now, there are various resources that are available on the main page that even you can access even without choosing a gene, such as these indel counts from the UK Biobank data. So as many of you know, and we will see that from Andrew's talk, the UK Biobank data was inputted into NOMAD. However, if you want to see a specific UK biobank counts for various variants, including indels, you can access that from here. That will give you an Excel sheet, which you can explore, and there are various notes about it. There is also an indel counts for the NDRS data. They're currently not displayed within CanVar. You have to open this extra sheet, and importantly, this is one of the resources that's only available if you are registered and logged in before you can open this sheet. And this is really important because it displays various indels that have been found in BRCA1 and BRCA2 and other cancer predisposition gene in the UK affected population, their counts and their ethnicity. Again, if you are looking for a missense variant, that's available on the main page, but if you're looking for an insertion deletion, that's only available from here, but it will give you the number of cases that have a specific variant. So these are affected cases, and the denominator within all ethnicities of the NHS data set or the white ethnicity as well. So you can choose the relevant one to be able to do case control analysis. If it's a missense variant, you can get that information from the main page, but if it's an insertion deletion, you can get it from here. So keep an eye on this if you are planning to use the CanVig guidelines during this scheme. So once you chose a gene and a variant, then you will have a large number of resources available directly using the Canva page. So the summary page is the first tab, gives you a lot of various information around the gene and the variant and the type of the variant, with some extra information available from other databases. And a really useful link here is the ClinVar status that you can see at the bottom right hand corner, because that helps you in PM5 interpretation. And so what you need to do is you need to click at the link that's available there, and then it will take a few seconds, but it will then do an assessment live from ClinVar and bring back the information on any variants that can be included within the PM5 count. So a valuable option that will give you a summary for PM5 count. Then if you move to the second tab, in silico predictions, there are lots of predictors and data available all together in one go. So you can see, for example, Rebel scores or base del, Rebel if you're using Canvic, base del if you're using Enigma cleansing guidance, and also Splice AI alongside many other predictors. So again, as for those who are familiar, you know, this is what people use a lot for PP3 assessment. Then moving next, you will have your case control frequencies. So the important one to highlight is the case frequency that's in that square. And again, this will only be available if you are registered and logged in. Otherwise, you will not be able to see this detailed display of the number of cases that have been detected in the UK, which is, again, as a reminder, a valuable resource for PS4 case control analysis. There's also different breakdowns of the nomad populations and the UK biobank populations. These are currently being updated, and the Canva team are working on making this even more inclusive and more flexible, with multiple other options to look at UK biobank data and the nomad subsets as well. Then if you move to genetic epidemiology, then especially for BRCA1 and BRCA2, you will have multiple large genetic epidemiology studies available here. So these are the studies that provide you with the multifactorial likelihoods for a variant being either pathogenic or benign. And not only data from these studies is available, but very helpfully, the Canva team have done all the conversions between likelihood ratio to exponent points. So the values that are on the right-hand side highlighted in yellow here are the ACMG LLR, which is essentially the calculated conversions of these scores to the exponent equivalence. So if you look at these values, you can convert them directly to the exponent scores under the correct evidence elements here. So for example, BP4 phenotypic specificity. So that's available. You can go there, you can take the points and directly use that within your variant classification. Next is the functional and splicing analysis. Again, a very helpful resource that's available within Canva. There's a slide missing here, I'm really sorry, but if you go back to the main Canva page that I showed, there is a link at the beginning next to the Canva sign that's called Canva UK datasets. And basically it summarizes all the functional studies and all the splicing assays and all the multifactorial multiple studies that are included within CANVAR. And this is really important because if you find a variant, if you find a study, like in this case, multiple studies have addressed this variant, then you will see the results. But if you don't see a variant, then it's worth checking, or if you don't see results for a variant that you're interested in, then it's worth checking what data sets have been explored. Because if there's a very recent functional study, it may have not yet been explored by the CANVAR team, and so the results from that study is not yet available. But what's really, again, helpful here is you can find a summary of the main functional studies or splicing assays, and also an assessment as per the Burnett et al. Criteria, so as per the PS3 clinician SVI recommendations. So someone from the CANVAR team would have looked through these studies, analyzed them, and then recommended a strength of evidence, whether that's PS3 strong or BS3 strong or moderate, and so on. And to be able to see all the summary of the functional studies, again, from the main page of Kanji and CANVAR, you can see, you can have a live link for all the CANVIG UK functional studies that have been scored as per the PS3 guidance. So this is the link. If you press on it, you will go to the live Excel, and you can see studies, for example, like Bowman or Finlay, which I'm sure most, if not all of you, are familiar with, and you can see what weight of evidence you can use them as. In this case, both studies, PS3 and BS3 at a strong level. There's also another very helpful resource available on the CANVAR website, which is the discussion forum. Again, this is only available if you're registered and logged in, but what's really helpful here is that you can start a discussion. You can suggest a variant classification, or you can recommend a change in variant classification. So if you reviewed a variant and you found you're trying to get some more information, or you found that some labs in the UK have listed this variant, but you can't see what classification they used, then you can actually submit a request on this forum, and all the CANVIG members will get a notification, and it starts a very nice discussion. So there are also a variety of levels of urgent or non-urgent requests that you can submit. So if you're really keen on some feedback from other users of the CANVAR or CANVIG community, you can put in an urgent forum chat, or there are other chats available. So what happens once you put something, once you propose a classification or ask for extra information or ask for classifications from other members of the CANVAR community, then you will start a discussion, people will get emails, and then they can log in and respond to that too. So that's all what I wanted to show within the CANVAR databases. I hope you found that really helpful. Please do explore it yourself, because it does hold a lot of really valuable information, not only from published databases, but also importantly from actual results from UK labs. Now I'm going to hand over to my colleague Andrew, who's going to take you through multiple other resources. Thank you.
Thank you, George. Next slide, please. So when we were asked to discuss different databases and resources we use for variant classification, the first thing that came to mind for me was the GNOMAD database. With the new recommendations coming out for gene-specific guidance using either GNOMAD 4.1 or 2.1 or 2.1 non-cancer, it got a little confusing as to what this all means and why you should use a particular database over the other. So I thought I'd go a little more in detail on what GNOMAD is. Next slide, please. So GNOMAD is a population database. It started in 2014 as the EXACT database, and then grew into the GNOMAD we now have. GNOMAD is composed of exome and genome data collected from around the world from over 100 different studies, including biobank submissions. The raw data is processed by Nomad using their bioinformatics pipeline, so all the data generated is consistent within a genome, within a version. The point of Nomad is to reflect the general population. This does include individuals with disease. So there are several case control studies included in the Nomad database. So this isn't a population of necessarily healthy individuals, it's a population of individuals. One thing they have actively tried to do is remove pediatric case control studies from their data set to remove severe pediatric disorders. That being said, the caveat with that is there are biobank submissions and not all of them are well curated as far as phenotype goes. So it is still possible to have individuals with even pediatric disease, although they have largely tried to remove this. Next slide, please. So there are a variety of different data sets available in Nomad, and I think this is where some people may get tripped up. They can roughly be broken down into two human genome assemblies, GRCH37 and GRCH38. In GRCH37, we have Nomad 2.1. This is over 140,000 individuals, of which 125 of these are exome data, and the remainder 15 is genome data. The next build, GRCH38, has version 3.1. This is comprised only of genomes, 76,000 genomes. Most of these have come from version 2.1, or some of them at least have come from version 2.1, as well as Nomad version 4.1. This is the largest data set, over 800,000 individuals. Once again, about 90% is exomes and a little over 10%, a little under 10% is genome data. So the data available in Nomad can be broken down by genetic ancestry. And this is what you can see on the right. What becomes pretty obvious is the European population is well overrepresented in the Nomad database, regardless of which version you look at. So for version 4.1, it comprises 77% of the individuals in Nomad. So just to be cautious when you're looking at the overall allele frequency of Nomad, the data is heavily skewed towards the European ancestry. That being said, there are improvements being made for the other ancestries. So when you're comparing Nomad version 2 versus version 4, you can see the South Asian and African populations have increased threefold in total number of alleles. They've also introduced in the newest version a Middle Eastern population. So other populations are being improved. Just be aware that it is heavily skewed towards the European population. Next slide, please.
Andrew, before you go on to the next slide, could I just put a reminder to the participants, please, to, if you have any questions for our speakers, we will handle the questions through the question and answer session at the end. So please just submit your questions through the Q&A at the bottom of your browser, your Zoom browser, and we'll pick them up afterwards through the discussion session with Andrew and George. Thanks, Andrew, and sorry for that.
Okay, so some other differences between the data sets. Version 4.1 is the largest data set currently available in Nomad. It can be subdivided into total and non-UK Biobank samples. So UK Biobank makes up a large proportion of these samples in version 4.1. And within version 4.1, you can select for a subpopulation that excludes the UK Biobank data. For version 3.1 and 2.1, they do not include the UK Biobank data. What you can do with these populations, though, is you can sub-select for cohorts of individuals, such as non-cancer or non-neurological, or only control in biobanks. And what this means is they will remove the cancer cases from any case control studies in that population. So you'll be looking at a smaller subset of individuals, but it will have known cancer cases removed from the population. They do this both for version 3.1 and 2.1, but have decided to no longer continue the service for version 4.1 as they felt the data set was becoming too large to accurately manage, which I think is completely reasonable. And next slide, please. So this is data from version 4.1. But as I said earlier, a lot of the data generated or in the NOMAD database comes from case control studies. This is the list of studies that compile the NOMAD database. So as you can see, the largest population is coming from biobanks and control sets. Some of these individuals aren't as well characterized, so it's hard to tell what type of phenotype the individuals may have, if any. Otherwise, they're coming from case control studies, largely either neurological or cardiac in nature. And they are including cases in the NOMAD database. So these are known affected individuals. In total, about 17% of individuals in the NOMAD database do have a disease or disorder. One thing that they have excluded from the NOMAD 4.1 database is cancer cases. So they have not included case control studies from cancer cases as they had in the previous versions. In version 2.1 and 3.1, they did include cancer studies such as the TCGA database, where they look at the germline variants in cancer patients and controls. This has been excluded from version 4.1. Next slide, please. So looking further at the NOMAD version 4.1, they've identified over 786 million SNVs. I think what the statistic they have is for every 3 base pairs, there are 2 SNVs in their database. The data is fairly evenly balanced between males and females, and they can also break down the age distribution of the individuals in the NOMAD database. Although all the age categories are covered, the majority of the data, both from the exome and genome data, is being derived from individuals between the age of 40 and 70. Next slide, please. Okay, so one very useful statistic that you can get from the NOMAD database is this group max filtering allele frequency. And why this is important is when evaluating a variant, if you've calculated or have been given a disease-specific threshold for that variant and you compare it to a filtering allele frequency, if the frequency of your allele is higher than the disease-specific variant, this would suggest that your allele is not compatible with disease and can be used as evidence either at BA1 or BS1. And so often what's being recommended is using the group max filtering allele frequency. What this specifically is the filtering allele frequency from the genetic ancestry with the highest allele frequency in NOMAD. And they calculate a 95% confidence interval and then display the lower boundary of this 95% confidence interval. In a couple slides, I'll show you exactly what this looks like. But it's the lower limit of the confidence interval, 95% confidence interval for the genetic ancestry with the highest allele frequency in NOMAD. And you can compare this to your disease threshold to determine if your allele frequency is higher than the disease allows for. Next slide, please. So when you go to NOMAD, there are a variety of ways of searching for variants in NOMAD. You can either search by gene or by transcript or by location. If you search by gene, this is what you get. You'll get the gene, the transcript, and the genome build. In the upper right, you can select which data set you want to look at. So this is where you select either version 4.1 or 3.1 or 2.1, or even a subset within those populations. In the middle, you'll see your constraint scores. This is where you'll find your PLI score and your look if you're using this. And then if you scroll down, you start seeing your variants of interest. Next slide, please. So your variants are sorted by position in the genome. There are a variety of different ways to filter for variants. So you can filter by loss of function or missense in-frame indels or synonymous. You can look at genome or exome data individually. If your variant is present in NOMAD, it'll be displayed using the HGBS consequence. If it's in a coding region, it'll be provided as the protein nomenclature. And if it's in a non-coding region, it'll provide the DNA nomenclature. If this variant is present in ClinVar, it will give the ClinVar classification for the variant. Additionally, you'll see the total allele count out of how many alleles were looked at that position and the allele frequency. And then you can click on an individual variant to get more information. Next slide, please. So when you click on a variant, you'll see the genomic position. You'll see the data broken down by exomes and genomes in total. You will get a total allele count out of an allele number, which then calculates an allele frequency. And then you'll see your group max filtering allele frequency. So this is the allele frequency from the genetic ancestry with the highest allele count, or not highest allele count, I'm sorry, highest allele frequency. And this is the lower 95% confidence interval of this. You can also see the number of homozygotes and the mean coverage for this position. On the top right, you'll see a variety of different resources you can click out to. So dbSNP, UCSC Genome Browser, as well as ClinVar and ClinGen if there is an entry. And then you can look at the different genetic ancestry groups and the individual frequencies within. If you click on the small arrows in front of the ancestry, it does break it down by male and female. Some of these ancestries also have additional subgroups within them, so you can further break down the populations. It gives the total allele count and the allele number, and then calculates an allele frequency for you. So allele count, the group with the highest allele count doesn't always correspond to the highest allele frequency. And this is because the European non-Finnish population has the largest total allele number looked at. So you can't just look at the allele count, you do have to use the allele frequency when determining which genetic ancestry has the highest frequency. And in this case, for this variant, the highest allele frequency is coming from the African population. You can see the allele frequency here is not the exact same number as the group max filtering allele frequency. There are two reasons for this. One is the group max filtering allele frequency is broken down by exomes and genomes, whereas here it's a combination of both. And additionally, this is the lower 95% confidence interval. So this is a conservative measure of the highest allele frequency. And so it will usually be a little lower than what the allele frequency is calculated for that ethnic group, or that genetic ancestry group. Next slide, please. If you scroll down further on a particular allele, what you'll find is some additional statistics. You'll get the age distribution, both for exome and genome data, of the total individuals and the individuals with this particular variant. You'll also get some quality scores such as read depth and additional quality metrics, which may be of interest to you. Next slide, please. And so I hope this has been a good overview of what NOMAD can and can't do for you. And ideally, it should help hopefully guide you as to which version of NOMAD you should be using and when it would be appropriate. If you wanted more information on NOMAD, here are a few links that come directly from the Broad Institute. There are videos to go into more in-depth information on NOMAD. Next slide, please. With my remaining time, I'm just going to go over the ClinVar and ClinGen databases, as these are also two really useful points of information. Next slide, please. So what is ClinVar? ClinVar is a database that has submissions for variant interpretations. ClinVar itself does not actually interpret variants. They just compile all the different submissions for a particular variant and make it publicly accessible. They are not the same as ClinGen, but they do work very closely with ClinGen in curating variants. ClinGen deals more with, well, ClinGen has a lot of different responsibilities, but in the context of variant interpretation, ClinGen is the group that is coming up with the refined variant interpretation recommendations, whereas ClinVar is the database of submissions of these variant interpretations. Next slide, please. So ClinVar has classifications for both somatic and germline variants. If you focus on the left, these are the germline classifications. You have the typical 5 you would expect: benign, likely benign, uncertain significance, likely pathogenic, and pathogenic. But on top of this, there are quite a number of other classifications you may see, such as likely pathogenic low penetrance, pathogenic low penetrance, as well as risk alleles, either uncertain, likely, or established risk allele. In addition, they have drug response and association. So there are more than just the typical 5 classifications from ACMG. On the somatic side, they have the AMP, ASCO-CAP tier 1 to 4. In addition, they have the VIC, oncogenic, likely oncogenic, uncertain significance, likely benign, and benign classifications. So just to be aware, ClinVar does classify both, or does have classifications for both germline and somatic classifications. Next slide, please. So I'm sure everyone who's been to ClinVar has seen the gold stars in the top left. And so just to clear up a misunderstanding of what these gold stars are, they aren't related to the classification of the variant. They are related to the review status of this. So 4 stars is the highest number of stars you can have. This is a submission from a practice guideline. I haven't actually seen any of these in ClinVar yet. What you are more likely to encounter are 3-star reviews, and these are reviews from an expert panel. So they are ranked quite high because it's a very reputable source. 2 stars are means there have been multiple submissions for this variant with evidence provided, and they're not contradicting. 1 star either means that there are multiple submissions, but they are contradicting, or there's only a single submission with evidence provided. And if there's no stars, it can mean either there's no evidence provided or no classification provided for the variant. If there are multiple submissions, but either a practice guideline or an expert panel has submitted a variant, that is a classification that will be displayed on top that will override any other classifications provided. Next slide, please. Okay, so there are several different ways of searching for a variant in ClinVar. You can use gene symbol, you can use HGBS, either looking at the transcript or the transcript and the CDOT nomenclature. You can combine the gene symbol with the CDOT nomenclature. You can search by location. In addition, you can also use a ClinVar accession number. You can also search within ClinVar within a range of a region of the genome. So it doesn't just have to be a specific gene. You can either narrow it down or broaden it to a general range using what they term either the UCSC style or nomad style chromosome coordinates. You can also search by disease. So if you type in a disease, it will come up with all the variants where a submission has been submitted to ClinVar under the context of that particular disease. Next slide, please. So if you search for a gene in ClinVar, in this example, it was BRCA1, you get a whole list of different variants. Usually the first few entries are larger genomic rearrangements, which encompass your gene of interest, and you need to scroll down further if you want to look for specific variants within the gene. There are a variety of ways of filtering your data if you are browsing a gene. You can filter by classification. You can filter by molecular consequence, so whether it's a missense or a frame shift or a splice site, variant type, variant size and length, I find to be quite useful if you are looking for a larger insertion or deletion. The breakpoints may not necessarily be well defined, so it might be challenging to look for a particularly large whole exonic deletion or duplication. So if you filter by variant size and variant length, it will narrow down your list of variants to browse. Next slide, please. So if you click on a particular variant, what pops up is on the top is the HGBS nomenclature, the classification, and the review status. So in this case, this is a 3-star review. This means this has been reviewed by an expert panel. Further down, you'll see when the first entry to ClinVar was, when the last evaluation was, and when the last submission was. You'll see the different alternate transcripts and the DNA nomenclature associated with these, and the protein nomenclature associated with these different transcripts. What can be useful is below this, they have alternative names for this variant. So for some historic variants that used to be called something slightly differently, they'll often list these alternative names down here. And this can be useful if you want to look in the literature for a variant and it has been studied for quite some time. You might find some older papers calling this variant a different name, and often you can find it here. There are also additional resources you can click out of. If there's a ClinGen entry, you can also get the dbSNP and Varzome from external links down here. Next slide, please. If you do continue to scroll down on this page, what you'll see is ClinGen dosage sensitivity curation. So if ClinGen has evaluated this gene for dosage sensitivity, it will provide a haploinsufficiency score as well as a triplo sensitivity score. And if it hasn't, then it won't provide this information. It also gives a list of the different conditions that submitting labs or submitting participants have classified this variant in the context of. So in this case, this was BRCA1. We're seeing a lot of hereditary cancer type syndromes. So breast cancer, or familial cancer of the breast, hereditary cancer predisposition syndrome, breast ovarian cancer familial susceptibility to type 1. This can be useful if your gene of interest either has both autosomal dominant and autosomal recessive disorders, or is associated with both autosomal dominant and recessive disorders, or 2 different disorders. It's useful to have an idea of what submitting participants classified this variant in the context of. Next slide, please. So if you scroll down further, you'll see the actual submissions into ClinVar. So on the far left, you'll see the classification provided. This was a 3-star review, so it came from an expert panel. In this case, it was Enigma in 2017. You'll see the condition that they classified the variants in the context of, more information about the submitter, and then more information. This is generally, when provided, it's the comments, the justification for how they arrived at the classification they did. So in this case, there have been several classifications, ranged from uncertain significance to benign, but because an expert panel reviewed this variant and classified it as benign, that is a classification that's being shown on the top of the page. One thing to point out, the expert panel classification overrules all other classifications, but the expert panel isn't necessarily using the most up-to-date classification. So for a variant like this where it's benign, it's very unlikely that it would change with new guidelines. But if you had a likely pathogenic or a VUS variant that was classified 10 years ago using older classification schemes, it may be like, you may want to take that with a grain of salt, because with the new guidelines, the evidence is refined, and therefore you may just be cautious with some of these variants that have been classified a long time ago, even if it were through an expert panel, because evidence may shift. Next slide, please. And so at the bottom of the entry for ClinVar, what you do see is if there's some functional evidence, it may be displayed over here. This isn't necessarily always up to date though. And then you get a list of the publications that were provided to ClinVar. Not all of these publications necessarily have your variant of interest. These are just publications that were data mined out of the ClinVar entry submissions. So some of these may be like, it's not uncommon to see the 2015 ACMG guidelines as a publication here. They may also have publications for variants associated with the genomic position, but not necessarily the exact same variant. So just be cautious when looking at these publications. They won't all necessarily contain your variant of interest. Next slide, please. And so now I'm going to shift to ClinGen. So that part was ClinVar, and that's the database of submissions for variants. And then this part, I'm just going to go over ClinGen only in the context of variant interpretation. ClinGen has a lot of information. It could probably take up a whole hour worth of talk on its own, but I'm not going to get into that. I'm just going to get into the useful tools of ClinGen for variant classification. So in 2015, ACMG published a variant interpretation recommendation. What became clear shortly after that is these guidelines needed to be refined and maybe more information provided to clarify some misunderstandings within these guidelines. And this is where ClinGen came into play. ClinGen works with expert panels to refine either gene-specific guidelines or general guideline rules. So you can have, they have sequence variant interpretation criteria, they have disease-specific expert panels, and criteria-specific registries. The criteria-specific registries, which you can find under the tools as well as in the working groups, is where you'll get your gene-specific recommendations. Next slide, please. And so this is the criteria-specific registry where you'll find gene-specific recommendations. They have them listed by working group. You can also search for your gene of interest. If your gene is being evaluated for gene-specific rules, they'll have a status, either classification in preparation or pilot rules in preparation. In these scenarios, you can't actually access the gene-specific rules, but if it has been released, this is where you can access the rules, and you would click on this link over here. And next slide, please. So this is an example for P10. This has been released. They are now on version 3.1, so they do update these guidelines. I learned the hard way, it's not the best idea to physically print these guidelines out because sometimes they change rather quickly. So I always go to this webpage to look for the most up-to-date version. This first link over here will link you to the most up-to-date version. It'll tell you when it was released. They often give an explanation of what the changes were. And then when you scroll down on the page, it will give you the full guidelines, and usually with supplemental information at the bottom. If you ever needed to go back to older guidelines, you can click on this third link over here, and this gives the history of the versions. So you can see for P10, it was first released in August of 2018. And then a little over a year later, they came up with the second version. And then a few years later, the third version, and now they're on 3.1, so their fourth version. There's some minor changes from the third version. And that is what's most readily accessible. Next slide, please. So generally speaking, if there is a gene-specific or disease-specific recommendation, I think it's best to probably use those as they'll be most accurate and specific for your gene of interest. However, if this doesn't exist, there are also general sequence variants specific recommendations. And so these are refinements of the 2015 ACMG criteria. This is where the first PBS1 decision tree kind of was published, as well as guidance on downgrading PM2 to supporting and the justification for that reasoning. There's a variety of other refinements that are also very useful. Many of the gene-specific guidance do follow this as well. But if you don't have gene-specific guidance available, this variant-specific guidance really enhances the ACMG 2015 guidelines. Next slide, please. And so that concludes my portion of this presentation. Thank you.
Thank you, Andrew and George, very much indeed for a very clear overview of the do's and don'ts and expert tricks and knowledge of how to actually get through variant classification databases and guidelines. So again, please, we've got lots of questions coming in on the Q&A function, so please do pop your question in. We've got some time now that we can spend running through them. We also have the Genie variant classification HRR genes about to launch. I think it's going live in April, so please do be on the lookout for that. You'll be notified if you've already registered. If you've already previously participated in one of the runs, you'll be notified. So Simon, will we start to work our way through some of the questions that have been submitted then? Yep,
we will do indeed, Sandi. Thank you very much indeed again to George and Andrew on that. I think you will agree from the presentations, there's potentially a very good resource for you and your staff training associated with competent, understanding the guidelines from this webinar. So please do share the webinar once we make it available on our YouTube channels in the next couple of weeks. George, I'm going to kick off with you. So really, this covers, I guess, your discussions on CanVar and CanVig. There are a couple of questions relating to accessing data, whether you have to be a registered user or not. I'll come to that in a second. But the first one is really, is there any restrictions on the availability of information on CanVar UK with respect to indels or small insertions and deletions?
Thank you, Simon. So they're currently not displayed in the same way as the other missense variants, and that's why if once they go back to the slides, they'll be able to see that there is an Excel. It's protected Excel. You can click on it, and you will be able to access the counts for the indels. So they're just displayed differently.
Okay, fabulous. I mean, I think. Yeah, it wasn't clear. It's not clear, is it, when you actually log on to actually be able to see that information? So that's great for clarifying that. The next thing really relates to the availability material of information depending on what status you have, whether you're a registered user or a non-registered user. Could you just clarify again what you can and can't see with respect to those two perspectives?
Thank you. So in order to see details of cases, so for example, if you have a missense variant that you're interested in order to be able to see if it was seen in the UK and the count and which labs have seen it in the UK, you have to be registered and logged in. Also, in order to be to take part in the chat, the forum, you have to be registered, and to be able to see the indel counts for so the cases, not the indel counts from the UK Biobank, but the indel counts for cases from the NDRS NHS data, then you also have to be logged in. Many people think that they have to be NHS users to be able to register and log in. No, as far as I know, the recommendation I checked with the CanVar team is to be from a diagnostic community. So anyone who works within diagnostic field can register to use CanVar.
Fab. So there's no restriction essentially on accessing it if you're a registered user, and that's I think the key thing, isn't it? What about the availability of information in CanVar itself? So is it restricted to the UK population, or is it broader than that? Could you just clarify, please?
So the cases that are listed, those are cases that have been screened within the UK. So the counts of cases that have been tested and found to carry a variant are from NHS data only resource.
Thank you.
Yes, thank you, Simon. George, just to follow on from that, I presume that's a longitudinal data set, so it's not just over the last sort of year or 24 months. It's actually got some depth to it. Is that right?
Yeah, it's historical data, goes back far from when we started testing, and that addresses one of the questions actually, because someone said what class is unaffected? Is it someone that had R207, R208? Well, the data goes far before we had test directory and specific R codes. So it's anyone who were deemed at the time that they got tested that they were eligible for testing. So almost all of them would have been affected.
Yeah, because the R codes were only introduced in 2018. So yeah, there's a vast amount of data there as well. Excellent. Andrew, I've got one for you around Nomad. So would you recommend using Nomad version 2 non-cancer dataset or version 4 as they've removed the cancer cohort for version 4? Which one would you recommend?
Well, it depends on the circumstance. So if there's gene-specific guidance, I would follow that. If there isn't, ideally, what would be provided for a threshold, or if you can calculate a disease threshold on your own using the Whiffen calculator and the CardioDB database. Version 4.1 is a much larger data set. And since they do have the cancer cases removed, I do think version 4.1 is much stronger. That being said, when you look at the disease-specific databases, some of them, the disease-specific guidance, some of them have been calibrated to version 2.1. So it's not necessarily easy to transfer that and scale it up to version 4.1. So if you have the option, I would use 4.1. It's much larger, but you can't necessarily in every case because it has been calibrated to a specific version.
Great, thank you very much. And then following on from that one, so can Nomad version 4.1 be used reliably to check for the frequency of large exon deletion and duplications?
That's a good question. I do use it. I don't know how reliable it is. Like the CNV data, the structural variant data isn't not nearly as in depth as the SNV and the small information. But you do have to take it with a grain of salt. So I would exercise caution, but it is, I think, more powerful than any other database I'm aware of for this. So it's at least a very good starting point.
Can I add?
Please, yeah.
Thanks, Andrew. If people go to the CanVig consensus guidance, we have a specific recommendation also on which database to use depending on the size of the CMV in question. So if it's, for example, has 3 contiguous exons or larger, then you can use Nomad CMV and Nomad SV. If it's smaller than that, we would recommend using Nomad SV. So there's some extra recommendations there if you are using CanVig guidance.
That's really helpful. Thank you, George. Simon, over to you for a few more questions.
Yeah, thank you, Sandi. So interesting. Well, I've got lots of questions coming in, so do keep your questions coming in as well. Interesting one here, which I think we can all relate to, which relates to the complexity of this whole process and maybe the knowledge associated with how you can submit your data. So one of the questions is really asking, so which I think it really applies to you, Andrew, is which working group or platform, I presume what we're really here is talking about a database, would you recommend as a simpler way to choose for a non-scientist, so say a clinician or a clinical geneticist, to share their data set? It can be quite difficult to navigate between them, particularly if you've not got any bioinformatics training, etc. Do you have any perspectives on that at all? So,
sorry, can you rephrase the question? I'm not.
So it really, I mean, this is a question, I presume it's coming from a non-clinical scientist perspective. So if you wanted to share information, you're choosing to query it as a clinician or a clinical geneticist, accessing the data can be quite difficult if you're not familiar with the details and nuances of each of the different databases. Do you have any perspectives on things you could do to easily access the relevant population data or frequencies associated with that? Is ClinVar, for example, or ClinGen an easy way of accessing that information?
ClinVar can be if there's an entry, but not every variant has been curated or added into ClinVar. Likewise, Nomad is useful, but not every variant is present in Nomad. It is really challenging. I mean,
Yes.
I'll be honest, in our
Yeah.
lab, many labs are using commercially available tools where they will link and compile all the data together for you. You can link out to it, but it is hard to find a one-stop shop where you would find all the information you need. Some of the common ones I would start with are ClinVar, ClinGen, Nomad. I actually signed up for CanVar UK, so although I'm not in the UK, I did sign up for it. I do find that's a very useful resource as well. Also publicly available. So I would start with those 4 places, but for freely available data sets, I'm not aware of one particular location where you can find all this information, unfortunately.
And of course, if you do have any specific queries relating to this, then you can always contact through the EMQN or GenQA directly, and we can try and help support the knowledge associated with that and put you in contact with the people who may be able to provide you some more detailed information. Sandi, over to you.
Yeah, thanks, Simon. So this one seems a bit complicated. A couple of people, it's coming in a couple of times. So what do you consider PP5 in pathogenic, 4-star or 3-star? So I presume that's in ClinVar.
Yes, so ClinGen has guidance not to use PP5 or BP. So I wouldn't use, this is where I think people may be misguided. I wouldn't use ClinVar as evidence for variant classification. I would use ClinVar as a source where to get your evidence from. So I wouldn't even use an expert review as a 4-star or 3-star as direct evidence itself. But it would be a good starting point to look for how they got to that classification.
Right, so great guidance there then. And I presume 2-star, there's another question coming in on 2-star, I presume exactly the same answer.
Exactly, yes.
Yeah. And then following on from that, it's also been flagged as important the date of review. And I think you touched on that yes, very much so, because guidelines change. But also the group who reviewed it in ClinVar, is that available? Is that something that should be taken into account? Or do you just, again, go back to it's a guidance and you should do your own classification?
I would always go back to, it's guidance and you should do your own classification.
Great. Thank you, Andrew, very much. George, I've just got one quick one from you. You keep talking about NDRS. I know what it means because it's quite a UK-focused organisation. Do you want to just explain NDRS and the value of it?
Yeah, thank you. So NDRS is actually the National Disease Registration Service, which is basically in the NHS, because we're all part of one bigger organization. Labs that are part of the NHS and with the NDRS, we have the ability and the facility to share all of our results. So we submit all of our data, so the variants that have passed clinical grade diagnostic testing, detected in patients that were eligible for testing in the UK, and because we are a publicly funded testing labs, patients usually have to align with certain criteria, might change over time, but with certain criteria. So all of this data gets submitted to the National Disease Registration Service, which they compile the data sets together, both for germline and somatic. And for the germline at the moment, they are then inputted into CanVar, and that's why we have this nice case control or case set that we can use a lot in supporting with variant interpretation.
Great, thank you, George. That's very clear. Simon?
Thanks, Sandi. So got a nice one here. I think this will appeal to you, George. So it's really about the discordances between ClinGen VSEP guidelines, gene-specific guidelines, and the CanVig gene-specific guidelines. I know you touched upon this in your presentation. This question is acknowledges that, but there's really asking which one would you recommend? So would you agree on mixing guidelines and taking criteria from one and criteria from another? What's your perspective on that?
Thank you, Simon. So I would definitely recommend against mixing guidelines. There are certain modifications that happen within guidelines that account for more than one evidence criteria at the same time. So for example, PM2, some guidelines with cap it at supporting, while other guidelines will allow it up to moderate. That's usually calibrated with other evidence criteria. So it's not when we change criteria and levels of strength of evidence that can be used, we take that in view of the whole set of guidance. So I would definitely recommend against using cherry picking bits and pieces. Why there are the differences, we highlighted that. What I would recommend is to choose a guidance that fits with local or national view. If you are an independent lab, so I would look at all guidance, review them, and then make a decision. And consistency is a key here, because you're making that decision based on a specific guidance and the set of evidence available at the time of use. We do have active conversation between Canvig and ClinGen. For example, we're actively discussing the Insight MMR guidance in order to try and align as much as possible. So we recognize this, we know that it's a problem, and we do our best in order to reduce differences between guidelines.
Thanks, George. So I guess the take-home message, as you say, is don't mix and match. Be consistent internally in how you actually use these guidelines, and make sure you consult the guidelines, particularly with the respect to the differences, and ensure you can stick consistently to one or the other. Okay, thank you. Sandi?
Yeah, a couple more about NOMAD, I'm afraid, Andrew. What's the difference between exomes and genomes in NOMAD?
Good question. So genomes will go further into the non-coding regions. Beyond that, I know the quality scores will, well, not the quality scores, the read depth will decrease a little bit. But within coding regions of a gene, I don't think there's a significant difference between the two different data sets, other than exomes tend to make up about 90% of what is in NOMAD, and genomes is around 10%. But if you're looking within the coding regions of a gene, I don't think there is a big difference.
Yeah, excellent. And that adds the value of the data set, isn't it? The more information in there. Thank you for that. Also, another question, is there a way to identify cases and the controls in NOMAD version 4.1?
No, they've removed that. So for version 4.1, they felt their data set was too large to manage this function. So they removed it after version 3.1. That being said, they have removed the cancer case control studies. So if you're looking at cancer specifically, they have excluded the case control studies. So in a way, it's sort of equivalent to the 2.1 non-cancer and 3.1 non-cancer, and that they've actively removed all the cases for cancer.
Right. Okay, great. Thank you. And then finally, another one that's quite linked to that as well. So for allele frequency determination in NOMAD, version 2.1 non-cancer is generally used. But in the case of version 4, which is required, for example, by the guidelines for MMR genes, the non-cancer subgroup doesn't exist. Is that correct? Somebody's just wanted clarification.
That is correct. But there are no cancer studies. So the non-cancer subgroup doesn't exist, but they've also not included the cancer studies. So it should be roughly equivalent.
Great, thank you. That's clear. Thank you. Simon, there's a couple of very general variant classification questions coming in. Do we want to tackle these ones now?
We could go for something that's slightly easier, maybe PM2. Anyone want PM2? So I'm going to throw it open to both of you. So can we get rid of PM2 at moderate level once and for all?
We can say PM2 altogether is going moderate and supporting once and for all, once the ACMG version 4 gets released formally. And then we all have to catch up.
That's wonderful. I think that's a
Fantastic.
great outcome. Thank you.
Do you want to go PM3 as well then?
Go PM3, yeah, go for it.
PM3? Okay, so how would you grade PM3? So this question is really asking for clarification, I think, really. Different levels of supporting, I mean, give different levels of such as supporting, moderate or strong, or would you consider the number of compound heterozygotes in the literature, and how would you count them?
So if you don't mind, I can start with this, George.
Yeah, of course.
ClinGen does have a recommendation for use of PM3. They break down what the variant has been found with. Has it been found confirmed in trans versus assumed? The classification of the other variant, whether it's a VUS, a likely pathogenic or pathogenic. So yeah, on the ClinGen website, they do have specific guidance for how to use PM3, which I think is very useful.
Thank you, Andrew. Sandi.
Another general one, what do you think of platforms like Barsome or Franklin? Are they useful tools for variant curation? What's your thoughts? Do you use either of those? Have you heard of either of those?
I can give a general recommendation, not specifically in reference to any of these tools. Just be wary with a lot of the cancer genes, because we have cancer-specific cancer gene guidance, we have gene-specific guidance. Most, if not all, available tools use generic ACMG guidance in their classification. So even for some of them, PP5 is an option to use. So I'll be just very wary between the difference of general guidance-based classifications and gene-specific. And there is a lot of published evidence on MMR, on BRCA1, BRCA2, that using gene-specific guidance gives you a better answer than using standard generic guidance.
Thank you, George. I think that's really wise, isn't it? You never know what, how up to date, or the data set that sits behind these tools, how accurate they are. So, thank you, Andrew. Any thoughts on that? Have you used any of these sort of curated platforms?
Sorry, I do agree with George. I think there are a number of commercially available tools that are useful for compiling evidence, but I agree with George that you should still be doing your own variant and classification yourself. But there are, I don't want to endorse one particular tool, but there are a number of tools that are very useful for compiling this evidence.
Great, thank you. Yeah, and it reduces the time, the hands-on time spent for your clinical scientist or whoever's classifying. Yeah, thank you. Another question about, now I know, George, you said to not mix and match guidelines. Another one here. So is it fine to use different versions of NOMAD for different metrics? And they've given an example. So allele frequency from version 4.1, but use version 2.1 for regional constraint scores. So again, a mix match question. Andrew, do you want to answer that for us?
That's a tough one. I don't recall ever mixing and matching for a particular variant. I do think for consistency's sake, it would likely be best to stay within a version, but it's not something I've ever encountered myself, if I'm being honest with you.
Okay, yeah, thank you. Simon, a few more left?
Yeah, have we got time for one more?
I think we have, yes.
Yeah, okie dokie. So we've got here one about BRCA1 and 2, so missense variant. So for a missense variant located outside any functional domain in BRCA1 or BRCA2, according to ClinGen, we could apply BP1 strong, which would be sufficient to classify it as likely benign. However, according to ClinGen's latest recommendations, BP1 supporting should be applied instead, meaning that an additional benign criterion would be needed to classify the variant as likely benign. Would computational predictors be enough to answer to address that? Who would like to answer that?
I think that would be double dipping in evidence, because I think they use computational predictors in part to justify using BP1 in this scenario. So I think you would need to find a different source of evidence. It goes back to what George was saying earlier about the rationale for the strengths of evidence within a guideline are all interconnected with different pieces of evidence. And so I think the in silico prediction guided allowing the use of BP1 for missense variants outside of key domains. And so you wouldn't want to then again use computational evidence on top of that. You'd want to find a different source.
Yeah, I would agree with that, Andrew. And I think we've actually covered that scenario at least once in some of our previous EQAs on BRCA1 and BRCA2, the variant interpretation. So we've specifically addressed that issue in those EQAs.
I think it will be in the next round as well. Some of the variants will address that question again.
So it's important that we obviously get it out there and inform you guys about that issue. Sandi, any more?
Yeah, I've got one here. Can we use PM5 for a known pathogenic variant with a ClinVar entry when there's another pathogenic variant at that position, or the variant of interest needs to be novel? Do you want me to repeat that? So can we use PM5 for a known pathogenic variant and it's in ClinVar when there's another pathogenic at that position, or the variant of interest needs to be novel? So can you use it when there's one already in ClinVar or it's never been seen before?
I'm not sure I totally... Do you understand the question, Andrew?
I think, yes. I don't think the variant needs to be novel. I think if it's already in ClinVar, that is acceptable.
Efficient.
But what would we say is we always recommend that if you're using a variant to provide information for PM5, don't, again, same as general guidance, don't use it off the shelf of what ClinVar is telling you it is. You need to also locally classify that variant using your guidance or the guidance that you're following before you can use it for PM5 for your variant under investigation.
That's correct.
Yeah. Thank you. And I think that is really the take-home message from today's webinar, isn't it? We've got these great tools and data sets available, but really they are, as I say, tools to be able to use your own knowledge and the guidance out there to be able to come to your own classification.
Thank you. Thank you. I think we're going to bring it to an end there. A big thank you to George and Andrew for all their support in helping provide this webinar to you. A big thank you to the AstraZeneca and MSD teams, obviously, for supporting it as well. An enormous thank you to you, the attendees, for joining us. Please do make sure that you keep an eye out for the email we'll be sending around from both GenQA and EMQN to announce the availability of the video. You know, it's a great resource. Utilize it for your staff training if necessary. And finally, a thank you to my co-chair, Sandi Deans. And we'll look forward to catching you at the next webinar, which I think is in May, if I remember rightly, and will be resulting from the variant classification EQA that's going to go out next month. Okay, have a good rest of the day, and we'll hopefully see you at the next webinar.
Thank you, everyone. Bye-bye.
Bye for now.